The science behind reproductive cloning, from DNA to cloned organism. Title: The science behind reproductive cloning, from DNA to cloned organism. Introduction: 100 words. Reproductive cloning, a technique that emerged in the late 20th century, has fascinated scientists and the public alike. This groundbreaking process allows the creation of genetically identical organisms through somatic cell nuclear transfer, SCNT. The journey from DNA to a fully developed cloned organism involves a series of intricate scientific steps. In this article, we will delve into the science behind reproductive cloning, exploring the fundamental principles, techniques, and potential applications. Understanding the basics of reproductive cloning, 300 words. Reproductive cloning is the process of creating an organism with identical genetic material to an existing one. The method most commonly employed is somatic cell nuclear transfer, SCNT, which involves replacing the nucleus of an egg cell with the nucleus of a donor cell. This donor nucleus contains the complete DNA of the organism to be cloned. The A to Z of somatic cell nuclear transfer, 400 words. 1. Cell selection. The first step in SCNT involves selecting an appropriate donor cell from the organism to be cloned. Typically, somatic cells, such as skin or muscle cells, can be used. 2. Cell culture and preparation. The donor cell is cultured and prepared for nuclear transfer. It is kept in a controlled environment to maintain its viability. 3. Egg cell retrieval. The recipient egg cell is collected and its nucleus is removed. The egg cell provides the necessary scaffolding for the cloned organism's development. 4. Nuclear transfer. The nucleus of the donor cell, containing the complete DNA, is extracted and injected into the empty egg cell. The cell is stimulated to induce fusion of the transferred nucleus with the cytoplasm of the egg cell. 5. Embryo development and implantation. The reconstructed egg cell is then cultured in a lab, allowing it to progress through the stages of early development. Once a viable embryo is obtained, it can be implanted into the uterus of a surrogate mother to continue development. Challenges and ethical considerations, 400 words. 1. Efficiency and viability. One of the major challenges in reproductive cloning lies in the low success rates. Many of the reconstructed embryos fail to develop or result in abnormal offspring. Scientists continue to research and refine techniques to enhance success rates. 2. Genetic abnormalities. Reproductive cloning raises concerns about potential genetic abnormalities in cloned organisms. Evidence suggests increased rates of genetic disorders and shorter lifespans in cloned animals, although the exact reasons for this are not fully understood. 3. Ethical implications. Cloning has sparked substantial ethical debates surrounding issues such as human cloning, human-animal hybrids, and the potential commodification of cloned organisms. Careful consideration of these ethical concerns is vital as we navigate the future of this technology. 2. Biomedical research. Cloned animals serve as valuable models for studying genetic diseases and developing potential treatments. Humanized animal models can provide insights into human physiology and pathology. 3. Organ transplantation. The ability to clone organs could revolutionize the field of organ transplantation, potentially overcoming the shortage of donor organs. 4. Species revival. Cloning extinct species from preserved genetic material, known as to extinction, remains a contentious topic. While still largely theoretical, the possibility of reintroducing vanished species into their natural ecosystems is intriguing. The process of somatic cell nuclear transfer can create clones with identical DNA, but differences in gene expression can lead to variations in phenotype and development. 1. Epigenetic modifications. Epigenetic marks, such as DNA methylation and histone modifications, regulate gene expression by influencing how the DNA is packaged and accessed by cellular machinery. During the process of nuclear transfer, these epigenetic marks may not be fully reset, leading to differences in gene expression between the donor cell and the cloned embryo. 2. Reprogramming techniques. To overcome these epigenetic differences, scientists have developed reprogramming techniques. These techniques involve erasing and re-establishing the epigenetic marks of the donor cell, allowing for more accurate gene expression in the cloned embryo. However, reprogramming is still a complex and challenging process that requires further refinement. Studies have shown that epigenetic modifications can affect cellular differentiation, organ development, and even behavior in cloned animals. 4. Epigenetics and cloned organ health. Epigenetic abnormalities in cloned organisms may contribute to health issues and decreased lifespan observed in some clones. Understanding and mitigating these epigenetic differences is essential for improving the health and viability of cloned organisms. Overcoming reproductive cloning limitations, 400 words. Reproductive cloning is a powerful technology, but it is not without limitations. Addressing these limitations is critical for advancing the field and maximizing its potential. 1. Efficiency and success rates. Increasing the efficiency and success rates of reproductive cloning is a significant challenge. Researchers are continually exploring new techniques to enhance the development and viability of reconstructed embryos, such as optimizing culture conditions and improving cell selection methods. 2. Genetic diversity. Reproductive cloning produces genetically identical organisms, which can limit genetic diversity and introduce vulnerabilities to diseases and environmental factors. To overcome this limitation, strategies such as combining reproductive cloning with genetic variation techniques, like gene editing or genetic recombination, are being explored. 3. Ethical considerations and public perception. The ethical concerns surrounding reproductive cloning continue to shape public perception and acceptance of the technology. Engaging in open and transparent discussions about the benefits, risks, and ethical implications of reproductive cloning is crucial for fostering public understanding and support. 4. Regulatory frameworks. Establishing robust regulatory frameworks is essential for ensuring responsible and ethical use of reproductive cloning. Regulations should address concerns related to animal welfare, genetic modification, and potential human applications while allowing for scientific progress and innovation. The future of reproductive cloning, 400 words. As scientific knowledge and technological advancements continue to expand, the future of reproductive cloning holds various exciting possibilities. 1. Genetic engineering. Reproductive cloning can be combined with genetic engineering techniques to enhance the traits or capabilities of cloned organisms. By manipulating specific genes, scientists could potentially create organisms with improved resistance to diseases, increased productivity, or even enhanced physical abilities. 2. Human cloning. While human cloning remains highly controversial and ethically contentious, it is an area that continues to be explored.